Hi students, last class we discussed about transcription and genetic code. During transcription, the message stored in the DNA is transcribed into mRNA. mRNA carries the base sequences. These base sequences are later translated into specific proteins. But there should be a mechanism to read this codon and attach it to the specific amino acid. Because amino acids have no such structural specialities to read the codon uniquely. So, Francis Crick understood there should be an adapter molecule. It should read the codon as well as connect it to bind to the specific amino acid. Later studies revealed that it is tRNA molecule which reads the codon and binds to the specific amino acid. In the actual shape, tRNA appears like an inverted capital L, whereas in the secondary structure, it looks like a clover leaf. It looks like this. It has 4 arms. Okay. And a short arm is also there, and one end. This end, it is known as the anticodon loop. Okay, this end is known as the anticodon loop because it can read the codons of mRNA. For example, if the mRNA sequence is A, G, C, suppose, then it reads the specific codon. Okay, here the complementary base of A, that is U, here it is C, here it is G, and the corresponding amino acid attached here, and this end is known as the amino acid acceptor end. Okay, so one end is the anticodon loop which reads the codon of the mRNA. The other end, the opposite end, is known as the amino acid acceptor end. And this end, actually it is T psi C loop, and this is the D loop, this is the variable arm. These three loops, the details of these 3 loops you will study in higher classes, but here you have to remember these 2, anticodon loop and the amino acid acceptor. There are specific tRNAs for reading specific codon. There is an initiated tRNA which reads the start codon, but the matter is there is no tRNA for reading the stop codons. Okay, so it is the tRNA which is an adapter molecule. At one end it reads the codon and the other end it attached to the specific amino acid. Okay, and now we can move to the next important process, that is translation. What is translation? The process of polymerization of amino acids into polypeptide, or in simple words, the process of formation of proteins. You know, in a protein, that is in a polypeptide, many amino acids are there. These amino acids are joined with the help of which bond? Peptide bond. But the formation of peptide bond requires energy. So in the first step itself, the amino acids are activated in the presence of ATP and it is attached to the specific tRNA. And this process is known as amino acylation of tRNA or charging of tRNA. When 2 such charged tRNAs come close to each other, the peptide bond formation will happen and it results in the formation of a peptide. Okay. And you know, the cell organelle which is responsible for protein synthesis, or the cellular factory for protein synthesis, is ribosome. There are 2 subunits in a ribosome, a larger subunit and a smaller subunit. When the mRNA encounters the smaller subunit, then the process of translation begins. And there are 3 steps in translation: initiation, elongation and termination. During initiation, the initiated tRNA binds with the start codon. You know, the mRNA carries the base sequences. It has start codon as well as the stop codon. When the initiated tRNA binds with the start codon, the process of translation begins. Okay, that is the first step. After that, the ribosome moves from codon to codon. And at that time, new amino acids will be synthesized because the specific tRNAs are there. These tRNA read the codon and the corresponding amino acid will be synthesized. Okay, and there are 2 sites in the large subunit of the ribosome. One A site and one P site. A site binds with the amino acyl tRNA, charged tRNA. Okay, and the P site that binds with the tRNA which carries the growing polypeptide. So, more and more polypeptides are formed there. Amino acids are joined with the help of this peptide bond and a polypeptide, long polypeptide chain will be synthesized. This is known as the elongation. That is the second step. When it reaches the last part, at the end, there are release factors. There is no tRNA for reading the stop codon. The release factor binds with the stop codons and the process gets stopped. As a result, the fully synthesized long polypeptide chain is released from the ribosome. That step is known as the termination. So, initiation, elongation and termination. These are the 3 different steps of translation. I told the mRNA carries codons. Start codon is there, stop codon is there and many other codons are also there. But before the start codon in the 5' end and after the stop codon in the 3' end, there are certain sequences which are not translated. Such sequences are known as untranslated region. UTR, untranslated region. Okay, it is present before the start codon in the 5' end and after the stop codon in the 3' end. It increases the efficiency of translation process. Actually, it is not translated into amino acid or proteins. Okay, these are untranslated regions. These are all about the structure of tRNA as well as translation. The remaining part we can study in the next class. Thank you.